Hello students, welcome back to Nucleus Neet Academy, and I am Kiran Kumar, your biology NEET faculty. After many days I am before you with another interesting topic, that is lipids from the chapter Biomolecules. So without wasting any time, let's enter into the topic. What are lipids? Lipids are generally water insoluble molecules. They will not dissolve in water at all. Okay, and the word lipids is derived from a Greek word called as lipos. Lipos means fat in Greek language. Okay, here fat refers to animal fat, right? So I will give a general introduction to lipids before we enter into the topic that will make you to understand this particular topic very clearly. In day-to-day life, we will be using lipids for various purposes. For example, after having a bath, we will apply coconut oil to our hair to keep our hair healthy. Similarly, we will be applying oil to our skin to keep our skin healthy. So our skin naturally it will produce some oil. You know that? So that is called sebum. It is produced by sebaceous glands present in our skin, right? So in our house, we also use lipids in the form of vegetable oil for preparing different types of food items. For frying fish, for frying chicken, for frying vegetables, we will be using vegetable oil. And where do we get vegetable oil from? Vegetable oil is obtained from oil seeds. So this vegetable oil is also a kind of lipid. Okay. So in our house, in order to worship God, we will be lighting diyas. So what liquid we use in diya? We will use oil, vegetable oil, right? We will use sesame oil or mustard oil in diyas to light the lamp, right? So this lipids can also act as a wonderful fuel to produce a flame, right? So in olden days when electricity wasn't there, at that time people, they relied on this oil in order to produce light. Okay. So similarly, in our houses, when doors and windows make squeaking noises, then we'll have one wonderful idea, that is putting 2-3 drops of coconut oil in door hinges will completely stop that troublesome squeaking noises coming from doors and windows. And the bicycle which we ride to our school, sometimes the bicycle will make some troublesome noise, uneasy noises. So at that time what we'll do, we'll apply few drops of coconut oil, then our bicycle will roll very freely without any noise. Okay, like this we'll use these oils or lipids for different reasons in our day-to-day life. Okay, now how these living organisms are using the lipids in their body, we are going to learn about them in this chapter, biomolecules. So first of all, how these lipid molecules are formed? Lipid molecules are formed by a chemical process called as esterification. Esterification. What is esterification? It is a process by which two compounds, alcohol and fatty acids. Alcohol and fatty acids, two molecules come together by forming bond called as ester bond. So this process we call it as esterification. So because of esterification of alcohol with fatty acids, lipid molecules are formed. Okay, and lipids are present in which materials or compounds we can see these lipids. Lipids are present in all the oils, including the vegetable oil. Lipids are present in animal fat. The animals will store the fat under their skin in specialized tissue called adipose tissue. So that is called animal fat. Lipids are also present in butter, which is a tasty food material which we use in our kitchen, butter and ghee. Okay, and lipids are also present in beeswax. Lipids are also present in our ear wax called cerumen that will keep our ear canal healthy. And lipids are also present in the form of myelin sheath of neurons. It will act as a wonderful insulating material for transmission of nerve impulses. Okay, and lipids are also present in the sebum. Sebum is a oily substance which is secreted by sebaceous glands in our skin that will keep our skin healthy. And lipids are also present in cutin. Cutin is a waxy material produced by epidermal cells in the shoot system that is cuticle is nothing but cutin. It is a water impermeable material. Lipids are also present in suberin cell wall, present in the cork tissue. Suberin acts as another water impermeable material. So like this, few examples I have quoted here. These are all the different roles, some of the roles of lipids in living organisms. Still there are plenty of roles of lipids in living organisms. We will discuss in a moment. Okay. And lipids, these molecules are largely made up of three different elements: carbon, hydrogen and oxygen. Majorly they are made up of carbon, hydrogen and oxygen, three elements mostly present in the lipids. Now the most important function of lipids in animals, that is being an animal fat. So animals, here I have taken a bull as an example. Any type of animal, it may be human being, a bull or a buffalo, or a cat, a dog, leopard, tiger, lion, any animal it may be. Animals store fat under their skin in the form of animal fat in a specialized tissue called as adipose tissue. In a specialized tissue called as adipose tissue. Okay. So inside the body, around the visceral organs also fat will be stored. Okay. So that fat, visceral fat, we are not going to discuss, but here we are going to discuss about the fat which is present below the skin, that is called subcutaneous fat, which is stored in the form of adipose tissue. What is the role of animal fat simply in all the animals? First is shock absorber. Whenever an animal it meets with an accident, when it falls down, when it gets hit by any object such as a rock or a stone, at that time this thick layer of fat, it will act as a shock absorber. It will receive the shock from that hard object and it will protect the internal organs. It will protect the internal organs like a kidney, liver from the damage. So like this, the animal fat, it acts as a shock absorber protecting the internal organs which are sensitive. Okay. Next, animal fat will act as a reserve food material. Whenever the animal doesn't have any food to eat, then it will go into a starvation. In that situation, animal first, it will depend upon carbohydrate called as glycogen. So this glycogen will be stored in the liver. First the glycogen reserve food material will be used. Once glycogen is completely exhausted, then at that time the animal will depend upon the fat, animal fat which is stored below its skin. Okay, as a reserve food material, animal fat will be used by animal afterwards. So even if animal fat is also completely used up, then the animal, it depends upon the protein which is stored in its body. Okay, so in this way the animal fat will act as a reserve food material during the time of starvation, right? So especially in the polar regions, animals like polar bear, that doesn't have anything to eat during harsh winter season, they go into a state called as hibernation. At that time they completely sleep throughout the winter. During that moment, this animal fat will come to use for maintaining the metabolic functions. Next is insulating material. Animal fat will act as a wonderful insulating material. It protects the body from shivering cold during the winter season. So this fat, animal fat which is present below the skin, it has beautiful insulating properties. It will not allow the heat that is generated inside the body to dissipate out. So this thick layer of fat, it will hold the heat within. So thereby the animal can keep its body warm even during harsh winter. And that is how many polar animals like penguins, seals and polar bears, they survive during harsh winter seasons by using this animal fat which is acting as a insulating material. So we human beings, we also have layer of fat below our skin. So it will protect our body during winter season, but additionally we will use sweater and all other clothing material to get further insulating advantage. Understood the point? Now let us discuss more in detail about lipids. Students, now we will discuss components of lipids. Any type of lipid molecule you take, the lipids will have two components. The lipids are made up of alcohol and fatty acid. When alcohol and fatty acid combine together, then they give rise to a lipid molecule. How the combining process take place? By process called as esterification. Okay. So here we are going to discuss about fatty acids. What are fatty acids? Fatty acids, they are simply the end products of digestion of lipids, which you have already studied in the human digestion process. So when lipids gets digested in the human digestive system, then the simple molecules which are formed are called as fatty acids. The same molecules here we are discussing. Okay, the fatty acids, chemically they are carboxylic acids with general formula are COOH. COOH is a carboxylic acid, and there is one variable group capital R attached to carboxylic acid. So this is the general formula of any type of fatty acid. And what will be the substituent in the place of variable group capital R? In the place of capital R, there can be a hydrogen, okay, or there can be alkyl group like CH3, or they can be C2H5, or they can be higher number of CH2 groups. Many CH2 groups can be also present in the place of this variable group R. Okay, now let us see some examples of fatty acids. First example, palmitic acid. Palmitic acid is a 16 carbon containing fatty acid. It is found in animal fat. Okay, palmitic acid has formula CH3, CH2 taken 14 times. That means there are 14 number of CH2 groups, and there will be one carboxylic group COOH. Okay, another example, stearic acid. Stearic acid, it has 18 carbons in it. It is a 18 carbon containing fatty acid. Formula again see here, CH3, and there are 16 number of CH2 groups. CH2 taken 16 times and COOH. Okay. Next, another fatty acid which is present in the animals, arachidic acid, a 20 carbon containing fatty acid, CH3, CH2 18 times and COOH. Okay, let us see complete structure of a fatty acid, palmitic acid. Here I have drawn the complete chemical structure of palmitic acid. Okay, so this is the structure of palmitic acid, 16 carbon fatty acid. Look here, carboxylic group is there here, COOH. Here I have represented, and how many CH2 groups are there? 14 number of CH2 groups. Look here, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So 14 number of CH2 groups are present, right? And last to 14th CH2 group, there is one CH3 group attached in the last. So this entire chain of CH2 groups form a hydrocarbon chain. What is that chain called? It is called hydrocarbon chain. Hydrocarbon chain. Okay, it is called hydrocarbon chain. So this hydrocarbon chain, a lengthy chain formed by attachment of several CH2 groups, and there are bonds between the carbons here. So these hydrocarbon chains are generally water repelling in nature. Water repelling means what? Hydrophobic in nature. They are having water repulsion or water repelling nature. Okay, in fluid mosaic model of plasma membrane, already you have discussed about this molecule called as phospholipids. Don't you remember? A phospholipid molecule will have head. Okay, then what is this structures? These are called as tails. The tails are nothing but hydrocarbon chains. They are nothing but hydrocarbon chains. And what is the nature of these tails in a phospholipid molecule? Hydrophobic or hydrophilic? The tails are hydrophobic. Okay, and these tails are nothing but hydrocarbon chains. And what is the head? The head represents the alcohol, which is hydrophilic component of lipid molecule. Okay, So now let us move on to alcohols. Let us move on to alcohols. So here I am erasing this. You can copy down. Okay. In a lipid molecule, there is another component called as alcohol. The alcohol group, generally it is a glycerol. Trihydroxypropane is the chemical name. Glycerol. So what is the structure of glycerol? If you see the structure of glycerol, there will be 3 carbons. First carbon CH2 attached to OH group. Second carbon CH, OH. Third carbon CH2, OH. That's it. So this is the structure of glycerol, which is a alcohol. So when a glycerol alcohol gets esterified with any type of fatty acid, then it gives rise to a lipid molecule. Okay. So then where esterification process takes place, esterification process may take place on the first carbon or second carbon or third carbon. Okay. So now let us see how esterification process occurs. So here I am putting one line. So once again I am drawing here the structure of glycerol alcohol, CH2OH, CHOH, CH2OH. So this is the structure of glycerol. Now I am representing a fatty acid. Look here, COOH, R1 represents a fatty acid. So here when a fatty acid approaches OH group of a glycerol, then esterification reaction will take place. During this esterification reaction, a water molecule will be removed. A water molecule will be removed. A water molecule will be removed. After removal of a water molecule, ester bond will be formed. Now let us see how this ester bond is formed. Look here, CH2, O, carbon, double bond O, R1. Okay, so this is the fatty acid. It is attached to the first carbon by ester bond. This is the ester bond. Next, second carbon CH, OH. Third carbon CH2, OH. Okay, so now here this is the glycerol part and this is the fatty acid part. When glycerol part and fatty acid part both are linked by ester bond, then the resultant molecule we call it as lipid. So since there is only one fatty acid attached to this glycerol, we call this lipid as monoglyceride. We call this lipid as monoglyceride lipid. Monoglyceride lipid. Monoglyceride lipid means what? There is only one fatty acid attached to the glycerol alcohol, then it is called as monoglyceride. Suppose a second fatty acid is attached to the second carbon like this. Again there will be a removal of a water molecule. Again there will be removal of another water molecule, suppose like this. Another water molecule will be removed. Then what happens? Another ester bond will be formed. So second fatty acid will be linked like this. Like this, second fatty acid. Now it will be called as diglyceride. What it will be called? Diglyceride lipid. Diglyceride lipid. Similarly, there is one more carbon. Here also one more fatty acid may be attached like this. COOH, R3, third fatty acid. Here also a water molecule will be removed. Then what happens? The third lipid, the third fatty acid is esterified to the third carbon, resulting in the formation of triglyceride. What is this lipid will be called? This lipid will be called as triglyceride. Okay, so like this monoglycerides, diglycerides and triglycerides. 3 types of lipid molecules are possible by esterification process. Understood the concept? Copy down these students. Students, now we are going to discuss about compound lipid. What is a compound lipid? A lipid molecule, when it is attached with a phosphate group or a carbohydrate or a protein, then we call such lipids as compound lipids. Under compound lipids, we are going to discuss phospholipid. So from the name itself, you might have realized what is a phospholipid. Obviously, a lipid molecule containing a phosphate group can be called as phospholipid. So already you have studied fluid mosaic model of cell membrane, right? So a fluid mosaic model of cell membrane shows double membrane of phospholipids. So there is 2 layers of phospholipids here. This is a layer of phospholipids. This is another layer of phospholipids. In both the layers, what are the unit molecules called? These are called phospholipids. Do you remember? Yes. So exactly this molecule we are going to discuss right now. So we call these molecules present in the cell membrane as phospholipids. Chemically they are lecithin molecules, also called as phosphatidylcholine. The lecithin molecule which you see in the cell membrane can be also called as phosphatidylcholine. Now let us see the structure of a phospholipid lecithin which is present in the cell membrane. Okay, so just like any other lipids, our lecithin molecule will also have glycerol, CH2 attached to a fatty acid, C double bond O R1. Second carbon attached to another fatty acid, O C double bond O R2. Next, third carbon of glycerol, CH2 attached to phosphate group, P double bond O OH O. So this is the phosphate group. Okay, the third carbon of glycerol is attached to phosphate group. Phosphate group is attached to 2 CH2 groups. Okay, the last CH2 group is attached to a nitrogen. The nitrogen is attached to 3 CH3 groups. 3 CH3 groups. The resultant structure, this is the structure of lecithin. This is the structure of lecithin, a phospholipid. Where is the phosphate group? Here you can see, this is the phosphate group. So that's why we are calling this lipid molecule as phospholipid. Copy down this, students. Students, now we are going to discuss another type of lipids called as derived lipids. What are derived lipids? These are the lipids which are derived from either simple lipids or compound lipids. Under the derived lipids, we are going to discuss steroids. Steroids are derived lipids. They are made up of 4 fused carbon rings. They are made up of 4 fused carbon rings, and there will be a long hydrocarbon chain in the structure of steroid. Okay, so steroids, there are different types of steroids: cholesterol, stigmosterol, ergosterol. Cholesterol, it is present in animals. Stigmosterol, it is present in plants. Ergosterol is present in fungi. So here in our syllabus, in NEET syllabus, we are going to discuss about the steroid which is present in animals, that is cholesterol. The formula C27H46O. So there are 27 carbons, okay, and there is only 1 oxygen in this structure of cholesterol. Now let us see the structure of cholesterol steroid. This is a carbon ring. This is a carbon ring A. It is fused with another carbon ring B. it is fused with another carbon ring B. B carbon ring, it is fused with another carbon ring called as C. This is the third carbon ring, C carbon ring. C carbon ring is fused with fourth carbon ring D. This is fused with fourth carbon ring D. This is the fourth carbon ring. Now, the fourth carbon ring D is attached to hydrocarbon chain. A long hydrocarbon chain. Okay, now the resultant structure which you can see here, this is the structure of cholesterol. Here there will be 2 CH3 groups attached and there will be 1 OH group attached to a carbon ring. So this is the full-fledged structure of cholesterol molecule. Okay, so here you can see carbon ring A, carbon ring B, carbon ring C, carbon ring D. All these 4 rings, they are mutually fused with each other. So this ring structure we call it as cyclopentano. Cyclopentano phenanthine structure. So any type of steroid you take, there will be 4 rings attached with each other. So this 4 ring structure is common for all steroids. It is called as cyclopentano phenanthine structure. Okay, now what is the formula? C27H46. Wow. So now let us see some of the points, important points about cholesterol. Cholesterol, first of all, it is present in the animals. It is present in animals. Mainly it is one of the component in the cell membrane of animals. It increases the stability of cell membrane in animals. This point you can copy down in your book. Next, cholesterol in animals, it is a precursor of steroid hormones. The steroid hormones like testosterone, progesterone, these hormones will be synthesized from the cholesterol molecule. Okay. Next, cholesterol is also a precursor for synthesis of vitamin D, also called as calciferol. So vitamin D is very crucial for healthy bones. Vitamin D synthesis takes place naturally in our body when our skin is exposed to sunlight. Okay, at that time the cholesterol present in our body gets converted into vitamin D. Okay, so that's why it is also called as calciferol, another name of vitamin D. And cholesterol is also required for formation of bile salts in the liver. The 2 types of bile salts, biliverdin and bilirubin. Next, cholesterol is absent in vegetable oils. Cholesterol is not present in the plants. That's why obviously it is not present in the vegetable oils, but it is present in ghee, animal fat, egg yolk, this type of food products. And in our body, cholesterol is digested by enzyme called cholesterol esterase enzyme. Cholesterol gets digested by cholesterol esterase enzyme. Now, is cholesterol good for our body or bad? So you cannot say completely bad, you cannot say completely good. Okay. So there are 2 types of cholesterols, bad cholesterol and good cholesterol. High density lipoprotein cholesterol, HDL cholesterol, is good for our body, but LDL cholesterol is obviously bad for our body. Okay. But bad cholesterol, when it accumulates in the inner walls of blood vessels, then it causes a health issue called as atherosclerosis. So the blood vessels, when it gets blocked by this bad cholesterol, the blood cannot flow freely through the blood vessels. As a result, when blood flow is restricted, then heart may not get enough blood to pump, so heart stroke may occur. Okay, This type of complications may occur because of bad cholesterol. So one should be conscious about their diet. We should, as far as possible, we should avoid the fat from animals. Okay, so copy down, students. This is the structure of cholesterol. Students, now we'll discuss about the types of fatty acids. There are two types of fatty acids in the nature: saturated fatty acids and unsaturated fatty acids. What are saturated fatty acids? Those fatty acids which has no double bond in their hydrocarbon chain, they are called as saturated fatty acids. You can see here in the hydrocarbon chain there won't be any type of double bond. So because of that, the chain will be straight. So this type of fatty acids, they are called as saturated fatty acids. Whereas unsaturated fatty acids, they will have double bonds in their structure of hydrocarbon chain. You can see here there is a double bond. So because of double bond, the hydrocarbon chain will show a bend or a kink in their structure. Okay, this type of fatty acids, they are having a crooked structure. So such type of fatty acids, we are calling them as unsaturated fatty acids. Remember the difference, the main difference here is double bond. Saturated fatty acids have no double bonds, whereas unsaturated fatty acids, they have double bonds in their hydrocarbon chain. Main difference. Now let us see some of the properties, physical properties of these two types of fatty acids. Or saturated fatty acids, they are solids at room temperature. They are solids at room temperature. They will remain in solid state. But whereas unsaturated fatty acids, they are liquids at room temperature. They remain as liquid in room temperature, just like our vegetable oil. Take sunflower oil, olive oil, or you can take mustard oil. So these oils, they remain in a liquid state. Here students, I have brought groundnut oil from my kitchen in order to demonstrate you and explain you about unsaturated fatty acids. Okay, so here in my right hand I have groundnut oil, in my left hand I have water in a bottle. So I am going to add this oil to a bottle of water. Let's see what will happen. The moment I add oil, you can see here the oil is just floating on the surface of water. It is not even going into the water. From this, what you can say, oil is having less density when compared to water. That's why oil always floats on the top of water. Okay, if you try to shake this, the oil will not dissolve in the water. That means the lipids are insoluble in water. And in what state you can see this oil, in liquid state or solid state? Obviously the oil is in liquid state. Understood? So when any type of lipid is in liquid state at room temperature, it is understood that it is a unsaturated fatty acid. So all the unsaturated fatty acids, they will be liquids at room temperature. Then saturated fatty acids, they will be solids at room temperature. Okay, saturated fatty acids will have high melting point. Unsaturated fatty acids will have low melting point. And saturated fatty acids are animal in origin. They are present in animal fat, butter, ghee, which are all animal products. Okay. But unsaturated fatty acids, they are present in edible vegetable oils like groundnut oil, sesame oil, mustard oil, olive oil, etc. and also present in cod liver oil. Okay. Next, carbon chain has no double bonds. Just now we have discussed. Here carbon chain has double bonds, and that is the reason it is showing a kink or bend in the hydrocarbon chain. So they have a straight chain, they have a straight hydrocarbon chain, they have a kink or a bend in the chain. You can see here, this is the kink or the bend. Okay. Examples of saturated fatty acids, very important for your NEET examination. I have represented here the examples along with number of carbon atoms. Palmitic acid present in animal fat, 16 carbons are present. Stearic acid, 18 carbon fatty acid. arachidic acid, 20 carbon fatty acid. Okay. Next, let us move on to unsaturated fatty acid examples. Oleic acid, 18 carbon, linoleic acid, 18 carbon, linolenic acid, 18 carbon. There is a small difference between these two fatty acids. This is oleic, linolenic acid. Here there is linic, here it is lic. That is the difference. Okay. Both are 18 carbon fatty acids. Next is arachidonic acid, 20 carbon fatty acid. So here you should not confuse between arachidic acid and arachidonic acid. Donic acid, it is a unsaturated fatty acid. Arachidic acid, it is a saturated fatty acid. So this is the concept about saturated and fatty acids. The next class we will discuss about nucleic acids. Subscribe to my channel and also comment and also like this video. We'll join in the next session. Thank you.